In this revision session, we'll be looking at the specific immune response. Within that, we'll be looking at cell-mediated immunity and we'll be looking at humoral immunity. Before we talk about the specific response, I want to talk a little bit about the non-specific response, the primary non-specific defense against pathogens in humans. So within that, we're talking about physical barriers such as the skin and the mucous membranes. Also, not dealt with in this lecture, but very important, the blood clotting cascade and the inflammatory response. The inflammatory response is a reaction of the body in response to a disease, damage, or irritation. The inflammation does result in heat of the affected area, pain, swelling, loss of function and redness, and it's caused by cells within the affected area producing two key chemicals, which we'll discuss in a bit, histamines and cytokines. Histamines are organic nitrogen-based compounds released by mast cells in the inflammatory response. They cause the blood vessels to dilate, so increase blood supply, which increases the temperature and makes the conditions unfavourable for pathogens. But it also increases the permeability of the vessels, which allows plasma to leak into the surrounding tissue fluid. This will obviously allow white blood cells to get to infected or affected areas, and the result will be swelling or edema in the area. You may have heard of or even taken antihistamines, which are a group of drugs used to counteract the effect of histamine in allergic reactions. So cytokines are a really cool group of proteins released by cells, and they're involved in cell signaling. They message the rest of the body to indicate infection, and they will signal to phagocytes to visit the local site. We will look at some of these actions later in this lecture, but they also message the hypothalamus, which obviously we know controls the homeostasis, which will cause the fever part of the response, the immune response. Interleukins are a specific type of cytokines, and we'll refer to those a little bit later when we talk about immunity. So in the following slides, we're going to talk a little more about phagocytosis. Obviously, we mentioned cytokines and their involvement in this. They message the phagocytes and initiate the process. I also want to talk about these opsonins, which are really cool. They're kind of indicators that are stuck to the cell surface of pathogens to mark them out for phagocytosis. In fact, the word opsonin comes from the Greek to mean prepare for eating. The opsonins are generally going to be antibodies that are stuck to the cell surface, as I say. I will refer back to opsonins when we talk about humoral immunity a little bit later. So the white blood cells involved in phagocytosis are neutrophils and macrophages. The key distinctions between the two white blood cells are that neutrophils have a lobed nucleus and macrophages have your normal bog standard nucleus there. So the reason for the lobed nucleus allows the cells to squidge down, so it allows them to kind of move between tissue a lot easier. So that's a physical difference between the two cells. There is actually a functional difference as well between macrophages and neutrophils, and I will explain that in the following slide. Okay, so we've got a phagocyte here. It can even be a macrophage or a neutrophil. And we've got our pathogen over here. Now, our pathogen we've recognised as being a non-self cell due to the antigens on its cell surface here, or also these opsonins, which I think I referred to in the last slide as being part of the humoral immunity process, and I will deal with that later. So let's just bookmark that here, and we'll come back to it later. So the pathogen is recognised as non-self by the phagocyte, and it will bind to the phagocyte receptor sites here. The binding of the pathogen to the receptor sites initiates phagocytosis, which is the endocytosis of the pathogen here within the plasma membrane, and this will then envelop the pathogen. So the pinching off of the plasma membrane here creates something we'll call a phagosome. It's very similar to a vesicle, which we've visited in other lectures. It's just the content of this will be the pathogen. So we call this a phagosome in this process. The phagocytosis also creating this is similar to endocytosis in other lectures. It's just with this particular process, we call it phagocytosis and a phagosome, rather than endocytosis and a vesicle. The next part of the process is the fusing of lysosomes. If you remember this from term 1, we spoke about lysosomes containing little vesicles containing enzymes, digestive enzymes. So these lysosomes will fuse with the phagosome. The result of the fusing of the lysosome and the phagosome is something we will call a phagolysosome. And within this phagolysosome, we have the enzymes digesting the pathogen. So once the pathogen's been digested within the phagolysosome, we have two possible outcomes. If the white blood cell in the process, this process, is a neutrophil, then that would be the end of the story. The pathogen's been digested, and that's that. If, though, it is a macrophage, then this cell will do something quite different. It will present the antigens on its cell surface. The presentation of these antigens on a cell surface is brought about by the presence of major histocompatibility complex proteins found on the cell surface of macrophages. These proteins are specific to macrophages and they're brought about by a set of genes, major histocompatibility complex genes, which are switched on in macrophages. So the presence of these proteins here in the cell surface allow the presentation of the antigens from the pathogen. This cell is now called an antigen presenting cell. And as I said, it is specific to macrophages rather than neutrophils. And we'll talk about this a little bit later, antigen presenting cells a little bit later. Okay, so now we're going to move into talk about cell mediated immunity. It follows really nicely from what we've been discussing with the macrophages becoming antigen presenting cells here. It does actually link in, and I'll cover that in a bit. So cell-mediated immunity involves T lymphocytes, or T cells, which we might refer to them as being. They're called T cells because they mature in the thymus, so that's where the T comes from. The T cells involved in this process are T helper cells, T memory cells, T regulator cells, and T killer cells, the coolest cell in the body. Fact. Cell-mediated immunity is an immune response to self cells, specifically self cells that are changed in some way. Their cell surface antigens are most likely changed in some way. Now, this can either be through infection with a virus, or it can be that the cell has become a cancer cell. So the first part of cell-mediated immunity really begins where the last process finished, which is we have a macrophage here. Remember, a macrophage specifically, the white blood cell that become an antigen-presenting cell, and it will engulf through phagocytosis the self cell here, which has changed in some way, either through viral infection or having become a cancer cell. So we've just got the antigen presenting cell here, the macrophage, antigens presented on the cell surface. Okay, so we've got our first T cell here. We've got the T helper cell. The T helper cell has receptors on its cell surface which will recognize and bind and fit to the antigens of the antigen presenting cell. So the binding of the antigens on the cell surface of the T helper cell initiates a response from the cell which causes the production of many T cells through mitosis. So this binding initiates cell division. Okay, so there's quite a lot going on here. I'm going to talk about the bit down the bottom here to start off with there. So these cells that we've just seen produced here through mitosis, the T cells, these are called T killer cells. So these cells will produce a protein called perforin, which is really cool. It's a protein that will get within the cell membrane and puncture the cell membrane of the target cells. So once the target cells have been destroyed, some of these T cells will remain. Okay, they become T memory cells. These remain in your tissue and your lymph nodes and form part of your immunological memory. So if you are reinfected or we have another circumstance with these cells, our T memory cells will quickly clone to become T killer cells. Okay, and these T killer cells will then produce the perforin, which will destroy the cells. T killer cells will also produce interleukins, which we spoke about at the beginning. We're talking about cytokines. So these are proteins produced, and they will go out and message phagocytes. And the phagocytes also will come along, therefore, and deal with these cells. Okay, so they will deal them through phagocytosis. Moving on to humoral immunity. Humoral immunity is similar in some ways to cell-mediated immunity, but also very different in that it is a response to antigens that are detached from non-self cells. So we're not talking about a response to the whole cell, we're talking about the antigens that detach from it, and we're also talking now about non-self cells, so antigens from pathogens such as bacterium. So humoral immunity can also be in response to antigen presenting cells, but again, this is from detached antigens. And also it is the response of antibodies. So rather than perforin and T killer cells, we're talking about the production of antibodies. Humoral immunity involves B lymphocytes. Now you're born with about 10 million types of B lymphocytes, each with a specific antibody on its cell surface. So you've pretty much got an antibody that will fit any invading pathogen's antigens, albeit sometimes a little loosely, and depending on how well the fit is, will depend on your body's ability to respond. This is called your pre-immune antibody repertoire. So if your body is infected with a pathogen, you will have the antibodies which will bind to its specific antigens. Okay, so with humoral immunity, if you remember, we said it is in response to detached antigens from the cell surface of an invading pathogen. We have the B cell here, and we have the specific antibody to these antigens on its cell surface. The antibodies will bind to the antigens here, and we call this clonal selection. So this particular B cell with its specific antibody will attach to the specific antigen. Clonal selection. This B cell has been selected for. The binding of the antigens to the antibodies will cause the B cell to engulf the antigens. The antigens are processed within the B cell, and they are then presented on the cell surface. This B cell is now called an antigen presenting cell. It's presented the antigens. Okay, so the presented antigens will now be bound to a T helper cell. Okay, so I know this is humoral immunity and T helper cells we discussed in cell-mediated immunity, but they do cross over into this process as well. So the T helper cell comes in here. The T helper cell binds to the B cell, okay, and it's this binding which causes this B cell to go through mitosis, okay. So the B cell will go through cell division, through mitosis, and it will create more B cells. This bit we call clonal expansion. So the cells produced we call plasma cells. These cells produce the specific antibodies. So they're cloned from the correct B lymphocyte with the correct antibodies. Therefore, they'll be creating the correct antibodies to deal with more of these pathogens that are in the body. The plasma cells produce the specific antibodies, and they have two functions. One, the antibodies can just destroy the pathogen, the invading pathogens. Two, they can also act as opsonins. They'll bind to the cell surface of the pathogens. They'll act as opsonins for the signaling to phagocytes to come in and to destroy the invading pathogens through phagocytosis. The T helper cells also produce interleukins, which we discussed in the beginning as being a type of cytokine. These interleukins will signal to other B cells which will come into the area. One final thing is that some of the B cells will hang around in your body, and they'll be remaining as B memory cells. So this forms part of your immunological memory. If you get reinfected at a later date, then these B memory cells will quickly go through clonal expansion, and they will produce a load of plasma cells really quickly, and you deal with the infection really, really quickly. This is being immune. This is immunity.